Okay, so now we're going to talk about meiotic cell division. I've got a list of terms, kind of important points over here on the side. I've got a little diagram behind me, and so I'm going to try to make sure that I hit all these points on the side and go through this diagram, and I'm going to try to do it in like 5 minutes. So, mitotic cell division. One of the most important parts about mitotic cell division is that it is usually involved with growth and repair. So it is not when we're making babies, right? So let's say you cut your hand, you got a blood and scab, and then you get new skin growing. That's going to be mitotic cell division. Or when you are a zygote that's one cell big and you are growing into a multicellular organism, that is going to be mitotic cell division as well. Basically, we're producing clones. Now, sometimes in that kind of division there are mistakes, and we make new cells that have slightly different characteristics, which is kind of the case in cancer and a couple other things, but most of the time we're getting a pretty close copy. So it's important that you know that we're not talking about sperm and egg here. We are not talking about generating a sperm. We are not talking about generating an egg. We are starting with a 2N cell, and if you don't remember what that is, watch the video on ploidy. And we're ending with a 2N cell, and those are exact copies. So I'm pretty sure I can check something off right now. Let's see. We got there we go. Growth and repair, not baby making. Okay, great. So, we also got this. Okay, so what's going to happen is a cell is going to receive a signal, and that's really, really important. This process does not start without the receiving of a signal, specifically called a mitogen, not a growth factor, which would make sense, but is not actually a growth factor. It's called a growth signal, and it's called a mitogen. So we're going to produce this cascade of events, which means something is going to bind to the surface of the cell. Hi, Trip.
Right.
Something's going to bind to the surface of the cell. It's going to start off something getting probably phosphorylated inside the cell, which is probably going to start some kind of transcription event. And then the cell is going to respond with gene expression and kind of head down this path. So, first things first, we start this process with G1. Most of the cells in your body are not doing this. Most of the cells in your body are differentiated to a degree, so they're serving as neurons or macrophages and gobbling up things. These types of cells, the cells that are capable of running this process, are some form of a stem cell, generally. Okay. So we start this with a signal. Yep. Okay, we start this process with a signal. And these stages, G1, S, G2, M, and cytokinesis, were named based off of how a cell looked under a microscope. So they don't make exact sense, but I'll go through them anyway. During G1, the cell is basically preparing for replication. That is the copying of DNA. So a lot of people think that the copying of DNA happens in mitosis. It does not. It happens in this step called synthesis. So in G1, the cell preps for that. It needs to make all the enzymes necessary to run cell division. It needs to make sure that all our helicases are bound to pieces of DNA and are ready to go ahead and copy, and they're going to try to recruit things like DNA polymerases, primases, ligases, and generate that large replication machinery. So after basically those machines become partially assembled, a signal is set out saying, okay, the cell is ready to start copying. It will then copy, so we have DNA polymerases copying long strands of DNA, and we'll have multiple DNA polymerases copying the same strand of DNA, kind of like multiple chapters of a book being copied at the same time. And that process will produce one exact copy of DNA. Not more, not less. It's kind of important that we know this. I lied about the not less. A lot of times when we copy DNA, we actually end up with a slightly shorter piece than we started with. The ends kind of fall off, and that's going to be important in cancer. If you ever learn about telomerases, that is the important piece. Okay, but the cell will signal the completion of replication. Once it signals the completion of replication, and that's a checkpoint, G2 begins. And in G2, the cell is going to start to organize and prepare itself to divide the DNA that it copies. I think of it as like ramen noodles. So you have a bowl of wet ramen noodles, and it's delicious. And you copy the ramen noodles, and it kind of just still looks like ramen noodles. In order to divide the two copies of the ramen noodles, what might be helpful is to condense the ramen noodles down into the dry form so they're kind of like bricks. And that is sort of what happens in mitosis. The DNA condenses and it becomes brick-like. It's held together by these proteins called condensin and cohesin. I didn't make those up. That's a real name. And that makes them easy to divide. You can see the difference between the beginning of mitosis and the end of mitosis is the presence of two nuclei. So here we're actually 2N. Here we are 2N. Here we are 2 times 2N. It's 2 times 2N and not 4N because actually 2 of those are copies. Here we are 2 times 2N. Here we are still 2 times 2N, but I'm going to notate that just a little bit differently because our nuclei are separated. And then in these cells, we are 2N again. After the division of the DNA and the reformation of the nucleus, so if you think about it, DNA is held in basically a sac called the nucleus. It's a highly, highly complicated sac, but it is still a sac. That sac breaks down, the DNA condenses, we pull apart the 2 genomes that were copied, and we've reformed the nuclei, and then we actually begin the process of splitting the cell. Now, cell splitting is really cool. A lot of it actually involves forming a sort of lasso around the middle of the cell and then kind of tightening that lasso until the cell splits off, and then you generate 2 cells. Interestingly, these cells have 2 different fates, usually. One of the cells will probably go back and be the stem cell so that this process can replicate, so that it can happen again. The other cell usually does something called differentiation, and we've been talking a lot about it, and specifically we've been talking about a lot of one of the mechanisms of differentiation, and that is epigenetic differentiation. So, because this cell will begin to receive signals telling it to become a neuron or a muscle or something along those lines, that this cell will begin to start making proteins specific to that type of cell type. So for example, this cell will start expressing only muscle cell genes. Or in this case, actually, neuronal cell genes. One thing that usually happens is a lot of this cell's DNA will become epigenetically modified. It will be shut down so that this cell will actually start to act in a way that this one doesn't. So at the end of this process, because of different proteins present in the cell and the fact that they're epigenetically different, you can give the same signal to this cell, and the cells will actually react differently even though they are clones. They have the exact same DNA, and that's a really, really important idea. Differentiation is the specific production of a certain subtype of genes, and that happens in response to different types of signals because of different proteins involved in the cell and epigenetic shutdown. So you might ask, well, these 2 cells actually look the same and they're probably experiencing the same signals. How do they become different? That's actually a super interesting experiment done by a guy named Bolavi. I don't remember his name. Anyway, he took 2 sea urchins and kind of watched how they split, and it turns out that one cell became one type and one cell became another, or one half became one cell type and the other half became another cell type over and over again. It turns out that when you split this cell, you split the insides of it differently, and based on what cell gets what parts of those insides, the cells will develop differently. Let's see if I hit everything here. We'll go over the checkpoints. We got sulfate. All right, so obviously you need a reason to know why we're studying this. It's like there's no point in studying this if there's no real application. And one of the major applications is cancer. So There are several checkpoints in this process. This process is highly, highly regulated. One of the first checkpoints is that this does not happen without a signal. One of the second checkpoints is that the DNA replication machinery needs to be assembled. Another checkpoint is that the DNA is copied once and completely copied. And then, during mitosis, what actually happens is two chromosomes will line up. The cell will shoot little microtubules across it and pull that DNA apart, those chromosomes apart. And that is also a checkpoint. The splitting of this DNA two ways and equally is a checkpoint. It's really, really cool how that happens. If you want to look that up, it involves a signal called MAD2. I don't know if there's a signal between mitosis and cytokinesis. I imagine there is. I just don't know it off the top of my head. So this cell process basically takes a lot for it to happen. And so it doesn't happen automatically, which is kind of a good thing. If you imagine your brain and your skull, it would be a bad thing for your neurons to start dividing rapidly inside of your skull. You would push out the neurons, they would literally squish, and you would die. So cancer involves failures of many of these checkpoints. For example, most cancers actually develop the ability to start replication without a signal. Sometimes they even get surrounding cells to produce that signal. So they become insensitive to growth signals, meaning that they don't need the growth signals anymore. And a lot of times they will have the surrounding cells produce growth signals. Once that happens, the cell starts proceeding down this pathway rather quickly. This part in here, kind of checking on the DNA, often involves a compound called P53, which is a protein that we've discussed several times. It's got that little positive part inside of it that binds the negatively charged DNA, and when that binds, it can help to signal or identify DNA damage, and if that happens, the cell stops. If there's a mutation in P53, which is actually common in about 50% of cancers, then the ability to check DNA becomes decreased, which means the number of mistakes that gets caught also decrease. As the number of mistakes that get caught decreases, the likelihood that mistakes will accumulate increases. Basically saying, if you destroy the thing checking the DNA, or disable the thing checking the DNA, then the number of mistakes in that DNA will accumulate and accumulate rapidly. That gives cancer kind of its ability, one of its abilities. It's called genomic instability. That is it for mitotic cell division. Really quickly, let's just kind of go over these big ideas again. We're starting with 2N, 2 genomes per cell. We're ending with 2 genomes per cell. We are not making babies. We are making growth, or we're not making growth. We are growthing. I don't care. We are growing or repairing. This process does not just happen like dominoes. It doesn't just happen one step, then another step, another step, another step. It happens like dominoes with a person holding the domino every third domino. So it's like domino, domino, domino. Are we okay? Are we ready to go? Okay, let's drop that next domino. Unless you have a mutation in one of those checkpoints, and that is common in diseases like cancer. The fate of the cells in mitotic cell division is generally one of two. It's going to be a stem cell fate, which means it's going to remain epigenetically unaltered or relatively epigenetically unaltered, or differentiated, which means that it will become epigenetically altered and therefore start responding to signals differently. For example, you apply the same signal to both of these genes, or to both of these cells, and if this cell has that gene shut down, it will not respond, whereas this cell will. This process, again, is highly controlled and starts with a signal. That's it for mitotic cell division. There are a ton of videos online that help to describe this process. I guess one last misconception that I think people have is that This process is meiosis, or mitosis, sorry. Cell division is not mitosis. Mitosis is an awesome process, and it happens in this process, but it is not the entire process. Mitosis is the splitting and organization of DNA, specifically dividing a copied set of DNA equally between two newly formed cells. That's it. I wish you all a good day and riches in the future, and